The last part of notes for Unit 11 is about the importance of bacteria. Prokaryotes are really important in helping clean up the environment because they are decomposers, and so they're definitely useful in terms of that, but they also do other kinds of things as well. There are prokaryotes that are useful for cleaning up contaminants because they have lots of nutritional diversity and they can form biofilms. Using organisms to remove pollutants from soil, air, or water is called bioremediation. So we're going to look at some examples of that and some other things that bacteria can do. First of all, here's a picture showing the treatment of an oil spill in Alaska. There are special bacteria that have been developed that eat oil. And so spraying a biofilm or a film of bacteria that eat oil over an oil spill, it can clean up the oil spill very, very easily without a lot of extra work. So bacteria are important for decomposing. They recycle nutrients in the ecosystems. Another important job that bacteria do for us is nitrogen fixation. They have the ability, or certain bacteria have the ability, to take nitrogen from the atmosphere and change it into nitrates or other kinds of nitrogen compounds that allows plants to incorporate nitrogen. Even though nitrogen is about 70% of our atmosphere, most organisms don't have the ability to use atmospheric or gaseous nitrogen to make their compounds. They have to use nitrogen compounds like nitrates and ammonia to incorporate those into their proteins and nucleic acids. Nitrogen-fixing bacteria allow this to happen. Another use or importance of bacteria is for genetic engineering, and we'll talk more about genetic engineering later on, but basically we can insert genes into different organisms using a plasmid that has been engineered with a certain gene. And then finally, bacteria can be pathogens or disease-causing bacteria, and they have a big impact on our world. Here's a picture of the root nodules in a plant that has nitrogen-fixing bacteria in the root nodules. These are called rhizobium species, and this gives this plant the ability to get the nitrogen compounds that it needs to make its necessary proteins and nucleic acids. This is streptomyces. Streptomyces is the source of a lot of antibiotics. Many of the antibiotics that you take when you have some kind of infection come originally from streptomyces. Anabina is a filamentous cyanobacterium. Cyanobacteria are photosynthetic bacteria, and you can see some specialized cells in the chain here. They're photosynthetic cells, but then there are certain cells that are nitrogen-fixing cells that allow the Anabina to incorporate nitrogen into the compounds that it makes. And here's Treponema pallidum, which is the spirochete that causes syphilis, which is a sexually transmitted disease and has caused lots of problems for hundreds of years. So some bacteria do cause disease. All organisms are exposed to various kinds of pathogenic bacteria. Not all of them catch the disease at the time. Sometimes you have some resistance to it. But most bacteria that cause diseases do so by producing some kind of a poison or a toxin. Exotoxins are proteins that the bacterial cells secrete into the environment. Endotoxins are components of the outer membrane in gram-negative bacteria that can cause illness as well. Here we have Staphylococcus aureus, a well-known skin and other kind of infection. This is a staph infection, and they produce exotoxins that are secreted by the cells, whereas Salmonella enteritis, which causes an intestinal problem, have endotoxins that are components of the plasmid membrane. The Staphylococcus is gram-positive, the Salmonella is gram-negative. We use something called Koch's postulates to prove that bacteria cause diseases. These are the 4 conditions that are used to establish that a certain bacterium is the cause of disease. These are the steps. You first of all have to find the bacterium in every case of the disease, that particular bacterium. Isolate that bacterium from a person or an organism that has the disease and grow it in pure culture so that you have cultures that are just that disease, that bacterium. Show that the cultured bacterium causes the disease when transferred to a healthy subject, and then be able to isolate the bacterium again from the experimentally infected subject. And we'll do an activity in class related to Koch's postulates so you can get an idea of how this works. They were used, they've been used for close to 200 years now, about 160 or so years. And one of the ways they were used most recently is to demonstrate that the bacterium Helicobacter pylori causes most ulcers. For a long time people thought that stomach ulcers were caused by other kinds of activity, overproduction of acid or something like that. But these two scientists, Barry Marshall and Robin Warren, proved that this bacterium caused that by actually ingesting some of the bacteria and producing ulcers in themselves. And they won the Nobel Prize in 2005. Here we have Barry Marshall and Robin Jones. So viruses, remember, are a box that has a genetic material in it, okay? And we oftentimes get viruses and vaccines mixed up. A vaccine is a preventive medication that you can take. It is made from weakened or killed forms of the virus or bacteria. And basically what vaccines do is they stimulate the body's immune system to recognize whatever that particular agent is as foreign, destroy it, and then remember it so it can destroy it again in future exposures. Antibiotics are drugs that kill infectious organisms, but they don't work on viruses because, again, viruses aren't organisms. A lot of the antibiotics are naturally occurring chemicals that come from soil-dwelling bacteria or fungi, like the streptomyces we saw earlier. Some bacteria develop resistance to an antibiotic and can survive exposure to it. That's called antibiotic resistance. And the fact that we use so many antibacterial products today makes antibiotic resistance even more prevalent than it used to be, because the bacteria have these naturally occurring abilities to be able to fight off or change things within themselves to avoid the exposure or the damage from the antibiotic. Most of the antibiotics deal with the cell wall, okay? So the cell walls, remember, are different between gram-positive and gram-negative bacteria. A lot of antibiotics don't work against gram-negative bacteria because their cell wall is not exposed. It's a smaller amount of peptidoglycan, and it's inside a slime layer, basically, that kind of protects it against most antibiotics. A lot of antibiotics target the ribosomes in bacteria, and if the bacteria doesn't have ribosomes, then it can't transcribe the proteins to make the cell walls. If it can't make those cell walls, then it can't grow, and it will therefore die. And so that's what the difference between antibiotics and vaccines. Remember, a vaccine can be from a bacterium or a virus, and it is used to prevent infection, whereas an antibiotic is used to treat an infection, and it only works on bacteria, not on viruses.